So in one of the recent videos that I posted, I mentioned the fact that neurons in the brain have some voltage, right? They use electricity to send signals, electron flow. Well, how much voltage is actually in a neuron? Well, we know it's roughly 70 millivolts at rest, which is, if you do the math, 0.07 volts. Well, how much is that in comparison to, a battery? Typical battery, 1.5 volts. And if you think about a car battery, right, those big batteries have roughly 12 volts. Okay, so 0.07 compared to 12 volts, that's not a lot, right, in that little neuron. But what you have to take into account is that you have roughly 86 billion neurons in your brain. So if you take 0.07 volts times 86 billion, roughly equal to 6 billion potential volts of electricity in your brain, right? Now, compare that to the 12-volt battery. Holy cow, right? In theory, you've got the potential for so much more voltage than a car battery. Well, what about something really big like a lightning strike? Well, we know lightning strikes are can be roughly 300 million volts. So, in theory, 6 billion potential volts in your brain versus 300 million volts in a lightning strike, the brain wins. Now, of course, this is theory, and being able to harness that, right, is near impossible. And I didn't mention things like resistance and ampage, and all of that comes into account as well. But just in theory, think about all that potential energy that it takes to run the brain. In this video, we're going to think about all that voltage in the brain and how neurons communicate. Let's get started. So, when we're talking about neurons, we want to talk about how a signal is transmitted. If you break it down into 3 easy steps, first we have to start the signal, propagate the signal, and then reset the system. So what's the starting of the signal? Well, if you think about neural transmission, there has to be some stimulus, right? I got to see something, I got to smell something, I got to hear something. It has to reach and trigger a stimulus. You can imagine a sound so weak that my ear didn't hear it, right? That didn't trigger a stimulus. But as someone turned up the volume, eventually my ear will hear it, send it to the brain, and I will have reached a stimulus. So first, we need to reach a stimulus, and that could be through sensory organs, it could be a thought in your brain. Once the stimulus is reached, that threshold, it sends it right down its neuron, maybe some other interneurons, and then eventually maybe we want a muscle to react. So this movement through different neurons or down a neuron's axon is the propagation of the signal, the wave moving through the neurons. And then once the signal gets where it's going, the whole system needs to be reset. So if you think of transmission of a signal into these 3 easy steps, I like to think of it as sort of dominoes, right? So if you have some dominoes, they're all standing like you see here. Well, if you take this first domino here, tip it over, what's going to happen? Well, of course, the rest of the dominoes will fall down if they're stacked appropriately. And this is a great analogy for how neurons work in the body. This domino here doesn't get up and move through the body, right? Just like this neuron here doesn't get and move to the muscle cell. But instead, a wave passes through them, just like dominoes. A wave will pass through these neurons to get to where we need it to go. Okay? So those are 3 easy steps of how a signal is transmitted. So now I'm going to give you a little more in-depth overview of how this actually happens. Action potentials will propagate impulses along a neuron. So what's an action potential, right? Think about the word. Potential to have some action, right? A little bit of a voltage sort of situation. Okay, there's some voltage like we talked about at the beginning. So membranes at rest are polarized. depolarized, just like a battery that we were talking about earlier, right? Okay, there's the battery. Sorry, my horrible diagram. You got a positive and a negative charge, right? In a battery, positive, negative. Well, your membrane at rest is also polarized. On the outside, positive. On the inside of the axon, negative, right? So you've got two poles, a positive and a negative, so it's polarized. Now, just like a battery, when we have a positive and a negative side, right, we know that this can do some work, right? The electrons are going to want to flow to the positive side, get some work done. Just like that in the neuron. What we'll find is that the movement of these particles will actually move a wave down. So when a stimulus is reached, right, you think a thought or you see something, you hear something, when we get that stimulus, these sodium and potassium channels in the neuron open, and then will cause the membrane to become depolarized at that location. So what we find is there are these little channels in the axon. Some are sodium channels, some are potassium channels. And when there is a stimulus received, these are voltage gated. So what happens is some of these charges will switch at that one location because it just moved in, right? And that will be depolarized at that location. Well, when one becomes depolarized, just like the dominoes, it starts a chain reaction, and this wave will move throughout the axon. And then at the end, like I mentioned earlier, we have to reset. So we'll need a pump, sodium potassium pump, which will use some ATP to get back to this membrane potential. Okay, so that is an overview of what I'm going to talk about in this video. We're going to go into more detail on each of these steps, but this is the overview of what we're going to be looking at. And there you can see that is the ATP-powered sodium potassium pump that's going to do the resetting. Okay, so let's start at the beginning. Basically, you have to understand that your cells are surrounded by a sea of charged ions. And you may remember from your chemistry class that we have anions and cations. So anions, these sort of negatively charged ones. Cations, we see the positive charged. Now, what we find is that even though there are negatives out here and there are positives inside the axon, the overall net charge is that inside is more negative, outside is more positive, right? So we're really concerned about the net. There are chlorines inside that have this negative charge. Sure, there's some chlorines outside, but we're more concerned about the net charge. So there's more concentrated anions inside, which means the net inside charge is negative. The net outside charge is positive because it has these potassium and sodium ions in the extracellular fluid. So right now we would say at rest this membrane is polarized because it's got the negative inside, the positive outside, just like a battery. There's my bad drawing of a battery again. Positive, negative. We have stored energy. We have the potential to have some action take place. Okay, so what then happens when you get a stimulated nerve or you have some threshold met? So this is an in-depth diagram, so I've zoomed in. So if you take one side of this neuron here, right, there it is, move it down, that's what we're looking at. So we're looking at the axon of one side of a neuron. So remember I told you outside is more positive, inside is more negative. So when a stimulus is reached, right, we reached the stimulus threshold, right, and at rest, remember I told you in the opening, it's roughly negative 70 millivolts or 0.07 volts. What happens is, if the threshold is met, these sodium channels in the membrane open, right? So it gets a little zzz, right? A little threshold is met. This sodium channel will open. Well, once a sodium channel is open, what do you think flows through? Yeah, the sodium, right? So the sodium ions will now diffuse into the cell. Okay, well, what has happened at this exact location now? Well, if the sodium, which is positive, has moved in, do you now see at this location those charges are reversed? So now the reverse charges at this point on the neuron means there's a positive inside and a negative outside at this one point. Okay? So we call that depolarization. It's no longer polarized at this exact location on this neuron. So what we see is going to happen next is that the cell becomes depolarized, and we're going to see a wave start to flow through, as I'll show you in the next diagram. So this is the action potential rising phase. So I have this little picture of a graph that I'm going to explain in more detail later, but just to show you where we were. So we were at the -70, that's at rest. If a threshold is reached, we're now going to see depolarization happening up to a positive 40 millivolts. So the wave impulse moves down. So remember that first sodium went through the channel. Well, because these are voltage gated, what's going to happen is you're going to see this next sodium channel is going to let it open and let its sodium in. And this will continue to happen. And then, of course, the next one, the sodium will come in until we get a wave down through the sodium channels. Well, now what's happened to this membrane potential? These sodiums have moved in, right? So now the charge is reversed. So we're up to 40 in the positive direction we get up here at this point. So now that we've reached depolarization, think of what has to happen. So these sodiums have moved in. And now, because these potassium channels are a little more slow than the sodium channels, the potassium channels will start to open. Okay, so what's going to happen now, look at these potassiums down here. Once the potassium channel opens, the potassium is going to fly out. So notice this is what we call the repolarization stage. As the potassium, not through that one, as the potassiums start to flow out, we're going to repolarize the membrane, which was just depolarized from the sodium. Okay, so there we go. There's some more potassium moving out. There's another potassium channel. There's that potassium, right? So the sodiums came in the depolarization. The potassiums moved out in the repolarization, and the charges are basically kind of reversed back. So now again, once you see, we've got more negatives inside and more positives outside. So we have essentially returned it to its original state that it was at the beginning. Now, what we will see here, do you see on this little graph a refractory? Sometimes more potassiums are pumped out than need to be. It's called a little bit of an overshoot. So it will go a little too much of the positive outside, but that will then return to a resting state, and the neuron can fire again. So speaking of resetting itself, after a neuron has fired, it has to reset. So we have all these sodiums that have moved inside. We've got these potassiums, right, that have moved outside. We need to get them back to where they were. Well, we're going to be moving against a concentration gradient. So anytime we move against, you know we're going to need our friend ATP, and we'll need a pump. The pump that we use is the sodium-potassium pump, as you see here. On the next slide, I'm going to show you exactly how that sodium-potassium pump works. So this is the active transport protein in the membrane. It's going to require some ATP. What we've got to do is we've got to pump out those 3 sodiums and then pump back in 2 potassiums. So here's how this works. So what we have here is, remember, we need the sodiums are in and they need to go out. So sodiums will come into this open space in this pump. And actually, there's a spot for 3 of them. We use some ATP. That phosphate group from ATP transfers down. And what we see is that the sodium-potassium pump is now phosphorylated. When it becomes phosphorylated with the phosphate group, it changes its structure, right? And opens up this other side, allowing, because of this conformational change, the sodiums to be released back to the outside of the axon. So now that it's out here, guess what's going to come in? These potassiums that are out and need to come in will come in and join here. And at this point, the phosphate group leaves, right? There it goes. And the pump returns to its original conformation, allowing for those potassiums to go back in. So what's the point of all of this? The sodium potassium pump is resetting the charge to what it was originally by pumping out the sodiums that came in during depolarization and pumping back in the potassiums from repolarization. Okay? A lot of words. I'm going to show you again on a graph what that looks like. So here's that action potential graph that we've kind of looked at throughout this entire video. And what you can see is at step 1, right here, step 1, we have a resting potential. And remember that resting potential is negative 70 millivolts. If a threshold is reached, which is up to roughly negative 55 millivolts, depolarization will occur. So remember, depolarization is when the sodium channels are open and the sodium rushes outside. The sodium channel opens and the sodium rushes inside the cell. Then what happens, of course, is the repolarization. The sodium channels are closed and the potassium channels open, allowing the potassium then to come in. So we can see that there. That's the repolarization, the reset. Often there's this overshoot like we talked about. Remember the potassium ions, often there's too many of them. So what we want to think about is this overall diagram, right? Do you understand what's happening? I'm going to review it one more time. Normally when your neuron is at rest, we're at negative 70 millivolts. Okay, that's resting. If a stimulus is reached, it causes the sodium channels to open and depolarization happens. Now, if we didn't reach negative 55, right, it was a sound that maybe you heard a little bit, but you weren't really sure of it, it didn't reach that threshold, right, it goes back down and nothing happens. We have to reach this threshold for depolarization to occur. Once the sodiums have moved, then we need the potassiums to move, right? The potassiums are going to repolarize. They're going to come into the cell, often creating an overshoot because they're so slow. Then we need to get back to resting stage. And then finally, of course, after all of this is happening, we need that sodium-potassium pump to pump back the sodium and potassium to their proper locations. Okay, so what happens when we get to the end, right? Well, I reviewed this in the last video, but I want to go over it one more time just to make sure you can complete the whole picture. So once all that stuff we just talked about happens here, okay, right? The threshold is met, the depolarization, the repolarization, the pump. When the signal gets all the way down to the end, the last domino is knocked down, there's a space, right? That space is called the synapse. That space is a barrier between this neuron and this neuron. So we need to get the signal, right? The signal started and it went down. We need to get it over to this neuron to continue. So it has to transverse that synaptic space. So even though the signal going down the neuron was electrical, the synaptic space we see is often chemical. Okay? And this could be either a stimulatory response we're wanting, or it could be an inhibitory, right? Either stimulating something to happen or inhibiting something from happening. And so this is, as we mentioned in the last video, a point where lots of weird things can happen. In fact, lots of mental health issues are because this synaptic space and this neurotransmitter reaction can be quite unreliable. So I want to review this one more time. All right, so what's happening at the chemical synapse? The action potential arrives, right? That's what we just talked about. All that voltage, the sodiums, the potassiums, they're moving, all that fun stuff. Okay, now it gets to the end. Once it gets down here, that voltage stimulates the calcium channel to open. So these calcium ions, which were outside the axon, right? This is the axon of the neuron we're looking at. Those calcium ions will come in, and that will stimulate this neurotransmitter vesicle to bind. Well, a neurotransmitter vesicle is a vesicle that has neurotransmitters in it, right? So once it binds, those neurotransmitters are going to be released into that synaptic space. They'll go across and find either another neuron or a muscle cell, and they will bind to the receptor protein. When that happens, if it's an ion-gated receptor protein, these ions will move into the cell and continue the signal moving forward. In the end, what happens to those neurotransmitters? Well, they can either be reabsorbed back into the cell, they can be absorbed by a glial cell. Lots of things can happen. And in this example, I'm showing you the axon of one neuron going to the dendrite of another neuron. But remember, this could also be a neuron that's a motor neuron attaching to a muscle tissue and maybe going to, say, muscle contract or something like that. So there could be lots of different examples where we would see this synaptic space. Okay, so this video, I understand, was quite a bit complicated. We were talking about, if you think big picture, we're talking about the fact that neurons in the brain, in the spinal cord, and the other peripheral neurons use voltage, use action potential to propagate signal down the neuron to get a signal transduced or moved to a different location. Then when it gets to the end of the neuron, at the synaptic space, we'll see neurotransmitters kick in. So big picture items, think neurons, voltage, electricity, synapse, think chemical. And this is so complicated, but it's fascinating to think that your brain, your response, your nervous system is all just these reactions of voltage and chemicals moving through the body.